Hey gang, we've got our section 2 of chapter 17 here, and this is going to end up being the details of transcription. Chapter 17 is transcription and translation, ultimately making a protein. And the quick goal of transcription is that we want to make a copy of the code from the DNA, and then that copy can be sent to the ribosomes where proteins are made, and the proteins don't have to deal with the DNA at all. It can leave it alone. So anyway, we have to make this copy. The copy is going to be called RNA. Ultimately, it's going to be called messenger RNA if it's going to be used to make a protein. So this slide is going to be an overview and some terminology that you'll hear, and then we will get into the details in the next couple slides. So first of all, transcription is going to be broken into 3 different steps. Step 1 here is the initiation step. Step 2 is elongation, and step 3 is termination, and we'll end up having a slide for each of those individually. But kind of the big picture here is we look at a big long DNA molecule, and that's what's represented in the blue here. Big long DNA molecule, and this thing would go on for, this might be 1,000 base pairs that we're looking at here, and this would go on for millions of base pairs. So this is just one little segment of DNA. It could be a couple thousand nucleotides or base pairs. Anyway, somewhere along here where there's a gene, and this dark blue region here is going to be a gene, there is going to be a area called a promoter sequence, and a promoter sequence, which we're looking at right here, promoter sequence, basically tells this is the place where transcription should start. Okay, the molecule that is responsible for making the RNA is called RNA polymerase. Should sound really familiar. It's very comparable to DNA polymerase that we learned about in chapter 16 when we were making DNA copies. So this is RNA polymerase, and it is very similar to the DNA polymerase, where as it builds a new molecule, it has to build by adding on to a 3' end, 3' open end. So it will always build in the 5' to 3' direction, and it'll continue building onto the 3' end, 3' end. That's just how DNA polymerase was. One difference here, this is great, one difference is that RNA polymerase has the ability to start from scratch. It doesn't need a primer at all like the DNA polymerase needed, so that's going to save us a couple steps in this whole transcription thing, that we don't need a primer. But initiation, we'll learn some details then, but basically RNA polymerase finds its spot to connect, and it does that by this promoter region, which is a sequence that tells it to start there, and then it's going to move. We're going to have this one moving in this direction, and there's 2 DNA strands here. It has to choose which one it's going to transcribe, and that the promoter region actually tells it. So we have the bottom one being the template. You can see this here. The bottom strand is going to be the template, and just some quick terminology. If the RNA polymerase is going to move this direction, then what they do is they call that like the flow downstream, kind of like water moving downstream. The RNA polymerase is going to move downstream. And then when they're talking about different parts of the gene, they'll say that something is downstream if it was to this end, or upstream of the promoter. And so it ends up the promoter is upstream of what's going to be transcribed. Another quick term for you, the part that's going to be transcribed is called the transcription unit. Right here, just terminology, transcription unit. Anyway, once the RNA polymerase has attached, and that is initiation, then elongation happens, and elongation basically is the building of our RNA. Okay, the building of our RNA on the template strand, and something that's going to be a little different from what we saw with replication is that as this RNA is built, you'll notice that our little bubble here, because we have to unzip this, that little bubble to get access to the nucleotides, it's a very small bubble, and as the RNA is being made, it's actually separating from the DNA so that the DNA re-zips. So wherever the RNA polymerase is, it's unzipping the DNA, bringing in new nucleotides to be complementary and to get this code, okay, and then it re-zips it as it goes back. All right, so that's, again, a little bit different from what we saw with replication. So this will continue down the length of the transcription unit until we get to an area called the termination region, or where there's a sequence that tells the whole process to stop. Okay, so that's the overview. We're going to look at more details on the initiation and the elongation and the termination here in the next couple slides. Okay, here's our diagram of the details of initiation of transcription. And first thing we're going to look at is this promoter region, which is shown in green right here, this promoter region. And that basically tells RNA polymerase where it should start, or the entire complex that we're going to learn about. It tells it where to start on the DNA. And it is a sequence, so this area, we're not getting any specifics on this area, but they do know that one part of the promoter region is something called a Tata box. The reason they call that is it's a lot of T's and A's and T's and A's, and you can see this here as A, T, A, T, 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 T. So lots of thymines and adenines. There's this Tata box, and that is one of the signals that says, hey, a gene is about to start. Okay. Now, RNA polymerase does not just go find this location, the promoter region, on its own. What it has is different proteins called transcription factors here. And some transcription factors are going to attach to this Tata region and the whole promoter region. And then they are going to actually help RNA polymerase attach to this location. Okay? So then ultimately we have these transcription factor proteins that are attaching. RNA polymerase attaches, and if we're looking at a eukaryote, which is what this whole thing is, if you look at a eukaryote, the specific type of RNA polymerase is called RNA polymerase II. There's actually 3 types of RNA polymerase, but RNA polymerase II is used for this. And then, after that is attached, there's other transcription factors that will attach afterwards. If you go onto the website in the animation section, I think I put up 3 animations on transcription. One of them really gets into the whole transcription factor thing. It does a good job of explaining what those are, so I'd encourage you to look at those animations. But anyway, once RNA polymerase has attached here, this entire thing, all the transcription factor proteins and the RNA polymerase II, that's called the transcription initiation complex. Transcription initiation complex. Once that has formed, then transcription can begin, and we can actually see this here. That part of the promoter region is the start spot on the for the transcription, and so we've already made our first couple RNA nucleotides, brought them in, and we're starting to make RNA. Other things to note, this promoter region will distinguish which of the 2 strands we use, and that's part of the sequencing that is in there. In this case, it's the bottom strand for this gene. In a different gene, it might be the top strand. Okay, it just totally depends. If it was the top strand, then our transcription would move this direction, but it's the bottom strand on this one, so we're going to move to the right. Okay, that's that. Now we can go to the next step, which is elongation. Alright, here's our diagram for elongation, and this is just zooming into the RNA polymerase right in here, RNA polymerase II. And again, if we're in a eukaryote, there'd be some extra proteins here that are making a complex that's moving along. But our RNA polymerase II, as you can see, this is great. RNA polymerase has the ability to do the unzipping of the DNA. There's no helicase needed for this, but it unzips the DNA, so you can see this happening here, unzipping the DNA. And it's just bringing in matching RNA nucleotides. So if there's a T on the DNA, it brings in an A. If there's an A, it brings in a U. If you remember, RNA has U's instead of T's. Yeah, it's just going to zip along doing this. I think the book says it might be 60 bases per second that it's going to be adding in here as it zips across here. But anyway, as it moves along, you'll notice that there's just a very small, and I mentioned this earlier, a very small bubble, transcription bubble that happens here. And then the RNA, as it's made, very quickly detaches from the DNA, so there'd be like this tail of new RNA being made out the back here, and then DNA can zip back together. Real quick comparison, this is the template strand on the bottom, the non-template strand on the top. If I look at the sequence of the non-template strand, okay, I can see that it is very similar to the RNA sequence. So I have a C and a C, A, T, E, and we can talk about that, but we'll say it's a U for now. A G with a C and a C. Sorry, they have this a little screwed up here. Should be 2 C's, because you wouldn't have a G and a G together. A and an A, and a T replaced with a U, T going to be replaced with a U. So a couple errors in there, but anyway, so that is elongation, and that will continue on until we get to our termination sequence, which we'll talk about on the next slide. Alright, for termination, there's not a really good diagram in the book to go with, so I'm just going to go back to this first overview picture where termination is described here at the end. And basically, just to point out that this bluish region right here would be a termination area. If this was a prokaryote, that termination area will have a sequence that as the sequence is transcribed, so meaning as we make the RNA that is the sequence, it will actually signal for the RNA polymerase to just completely detach, which is what we're seeing here. Okay, it just detaches, breaks off. We've got our RNA transcript, which can be used, and we're good to go. Okay, if it's a eukaryote, it's going to be a little bit different. So I'm going to try to delete all these things. With a prokaryote, there's still going to be a signal at the end here, okay, a termination signal. And there's a couple things. They don't know the entire signal, but they do know that part of it is called a polyadenylation signal. And I'm not going to write that because until I write that, it'll take a complete minute. But what that signal is it is A, U, A. The reason it's called polyadenylation is there's many A's, which are adenines, and so it's poly-A. Oftentimes they'll call it a poly-A signal, many A signal. But what that does is it actually, that and some other sequences that they're not quite sure what they are, basically signal for the RNA and some proteins that are going to be associated with it to reach back and cut off the RNA as the RNA polymerase, and so I'm going to do my best RNA polymerase thing, as the RNA polymerase continues to move down the DNA. Okay, so it's going to cut off the section that we want, and then RNA polymerase, it's almost like it's clueless. It has no idea that this just happened, and it just keeps working its way down the DNA, continuing to transcribe. So it'll actually transcribe hundreds and thousands, possibly, bases past what it was supposed to do, and so it's kind of a lot of wasted energy and nucleotides that don't need to be pulled together or being pulled together and whatever. But anyway, the section that we wanted, the transcription unit that we want, because of this polyadenylation signal, basically cuts itself away from this rest of this wasted transcription that is happening. But that's how it works in eukaryotes. It's much more complex. Almost seems like it's not as efficient at what it's doing, but that's the termination there. So what we have now is we have our RNA. So I have this circled down here a bunch of times. But we have this RNA that was made, and the next thing that's going to happen, we're going to kind of focus in on eukaryotes, and eukaryotes are going to have to do some processing to this RNA, some fixing, and put some little signal things on it and whatever, so that it can then go and be used to make a protein. So that's it for transcription, and the lights just went out in the room, so we'll see you later.